Techniques in animal biotechnology. There are three techniques in animal biotechnology: genetic engineering, cloning, and assisted reproductive technologies. First is the genetic engineering. Creating transgenic animals or recombinant DNA. Recombinant DNA technology involves the stable insertion of a foreign gene or DNA sequence into an animal's native genome. Its goal is to confer a new trait, produce a desired therapeutic protein or what we know as biofarming, or to create disease models. There are three techniques used to physically introduce the foreign DNA into the host cells. First is microinjection. It is where we are directly injecting DNA into the prenucleus of a fertilized egg. Second is the electroporation. It is done by using an electric pulse to temporarily open pores in the cell membrane. Lastly, viral vectors. It is where we are utilizing modified viruses to deliver the genetic payload. Next, precision genome modification. Gene editing, or CRISPR, is the most revolutionary tool allowing scientists to make specific targeted changes such as cuts, deletions, or insertions to an animal's DNA with unprecedented accuracy. An analogy of this is like molecular search and replace function. Gene knockout. It is a deliberate strategy to disable or inactivate a specific gene entirely. Its purpose is to study the loss of function, phenotype, and determine the gene's exact role in development or disease processes. It also enables rapid development of precise disease models and enhances desirable traits. Next, cloning. Somatic cell nuclear transfer, or SCNT. The SCNT mechanism, or the process, the donor DNA of the somatic cell is a nucleus that contains the complete 2N genome. Is taken from a differentiated body cell, for example, the skin or mammary gland of the animal, to be cloned. Then the host cell, which is an unfertilized egg cell, has its own nucleus removed, a process which we call enucleation. Then reconstruction. It is where the somatic cell nucleus is inserted into the enucleated host cell. Then activation. It is where an electric pulse or chemical treatment is applied to simulate fertilization, triggering the reprogrammed egg to start dividing like a normal embryo. Assisted reproductive technologies, or in short, ART. In vivo techniques, it is where we are maximizing elite genetics. Artificial insemination, it is the process of manually introducing semen, often from genetically superior males, into a female cervix or uterus to achieve fertilization. It is widely used in livestock, mostly cattle and swine, to rapidly disseminate desired traits across a population. The embryos are collected non-surgically from a genetically superior donor female and transferred into a less valuable surrogate. This allows lead females to produce many more offspring than naturally possible, maximizing their reproductive potential. Lastly, in vitro techniques, it is a lab-based fertilization. Ovum pickup and culture, or OPU, this technique involves retrieving immature eggs directly from a female's ovaries using ultrasound-guided aspiration. The eggs are then matured in vitro. In vitro fertilization, or the IVF, the matured eggs, often from OPU, are fertilized by sperm outside the body, typically in a petri dish in a laboratory setting. The process flow is that OPU provides the host cell, which are then used for IVF, to produce viable embryos for subsequent transfer, or PT. This process is essential for high-value animals as it enables embryo production without relying on the donor's natural estrous cycle.